In the last section, you will remember that we talked about the various levels of structures of protein. You can damage those levels of structure or those structures by doing things like exposing that protein to heat, in other words, when you're cooking, or change its pH, or do a couple of other things that means that the shape of the protein will change. The fancy word for changing the shape of a protein is denaturing it. So a denatured protein is a protein that shape has been changed. When you change the shape of a protein, you change its function. Let me repeat that so I know you've heard it at least twice, and you'll hear it more than that in this section. When you change the shape of a protein, you change its function. Reaches a point where the protein is damaged so badly, it loses its function. It's irreversibly damaged, and when it loses its function, when it loses its shape, it loses its function. When you eat a protein, whether you're eating something like beans and rice or peanut butter on wheat bread or a hamburger or a piece of cheese, the protein that you eat in that food does not immediately turn into a body protein. You actually have to break it down through digestion into amino acids, and then those amino acids have to be put back together at the ribosome into the primary structure, and that primary structure may or may not be changed to secondary, tertiary, or quaternary structures of proteins. There are going to be specific enzymes that will digest the protein that you've eaten, and others that will break down those long polypeptide chains into tripeptides or dipeptides, and then knock those down into amino acids. This is a great little reminder and review of the material that we went over when we were talking about the anatomy portion, so make sure you check that out. So again, when you denature a protein, you change its shape, so you're changing its function. What can actually do that? We mentioned changes in pH, either more acidic or more basic. Heavy metals, like silver nitrate, or inorganic salts, and organic solvents, like alcohol, for example, and of course, heat. So when you're cooking your meat, or you're scrambling your egg, you are changing the shape, so you're changing the protein's function. So here's a question. Can you actually use the amino acids from that protein that has been denatured and use it for body functions? Well, what do you think? I've actually already given you the answer. Yes, you can. You just need to make sure that the protein is broken down to the level of amino acids, and then those amino acids will be strung back together into the primary structure of a protein, which is a specific number and specific sequence of amino acids to form a polypeptide chain. So when you digest proteins, the first place that any action for protein digestion to occur is the stomach. You'll remember that the acidity of the stomach is quite high. Got hydrochloric acid in it. So sure enough, that acid, you know that changes in pH can denature proteins. So sure enough, that acid is going to denature proteins that you eat. So the digestive enzymes that need to break it down can do that. Now, that hydrochloric acid in your stomach is also going to be super important, because the stomach will produce an inactive enzyme, which is known as pepsinogen. The hydrochloric acid will convert that to the active form, which is known as pepsin. In general, you want the inactive form to be hanging out in your stomach, because if it was the active form, you would actually be breaking down any proteins that are associated with your stomach, including the ones that are on cell walls. So it's important that pepsinogen is inactive until you actually need to digest proteins. And then the hydrochloric acid will convert to pepsin. Once you're in the small intestine, you'll remember that the pancreas is kind of that hotbed of enzymatic release. So those pancreatic peptidases and proteinases, as well as proteases that the intestines actually produce and secrete themselves, will break them down, hydrolyze them even further into short peptide chains, into tripeptides, into dipeptides, and then down to the level of amino acids. A peptidase is an enzyme that is going to split the di and tripeptides into amino acids. Proteases actually break down larger proteins. And that shows you what's going on there. Proteases versus peptidases. So what happens once they're broken down? There are very specific active transport carriers that are made of protein, which will pull amino acids into the cells. It'll also bring in some of the dipeptides and tripeptides. Once those are moved into the intestinal cells, those amino acids actually may either be converted into glucose to be used for ATP, or they will synthesize compounds that are necessary for those intestinal cells to keep functioning. The amino acids that are not used by those intestinal cells will get moved off into the circulatory system, which will carry them to the liver for processing and use by other body cells. And that is how you get amino acids to every cell in the body. Make sure you watch that, and I'll catch you in the next section.